How do you switch off a gene in exactly one cell type and leave it running everywhere else in the body? Not delete it, just quietly silence it in exactly one tissue and nowhere else. That is the real question behind the Duffy-negative blood type. The obvious guess is that the gene itself must be broken or missing, but it isn't. The gene is completely intact. What's broken is something much smaller and much stranger. Researchers sequenced the gene itself in Duffy-positive and Duffy-negative people. There was no difference. The instructions for building the protein are identical, so they look just upstream at the promoter, which is the switch that turns the gene on. There, they found exactly one difference, a single letter at position -46. Duffy-positive people have a T, and Duffy-negative people have a C. That letter sits inside the docking site for a protein called GATA1, which is a master switch that turns genes on, specifically in developing red blood cells. So you swap that one letter and GATA1 can no longer grip that site. So no GATA1 grip, no erythroid on signal. Everywhere else in the body, kidney, spleen, blood vessel lining, GATA1 was never the one flipping this switch to begin with, so those tissues never noticed any difference. Only red blood cells depend on this exact doorway, and that's the only place the light goes out. Now, this is cool and all, but who cares? So what? Here's why a single silenced gene turned into one of the most common traits in an entire region of the world. The Duffy antigen isn't just sitting on red blood cells doing nothing. It's the actual docking receptor that Plasmodium vivax, one of the parasites that causes malaria, uses to break into red blood cells. The parasite can still touch the cell, it just can't get in. No receptor, no entry. That is a huge deal in a region where vivax malaria has been a constant, deadly pressure for a very long time. A mutation that happens to silence this receptor, specifically where the parasite needs it, would have been strongly favored. The paper's authors point out that the frequency of this trait in West Africa lines up closely with resistance to the parasite there. What's not fully settled is whether that's the mutation itself being favored, or the parasite adapting to populations where it was already common, or some mix of both. The paper leaves that open, and so do I. One thing worth being precise about here is that this is a protection at the population level, built up over an extremely long time. It's not a guarantee for any one person, and it doesn't mean people without the variant are defenseless. So, how common is this variant really? And this is where a single base pair from a 1995 paper turns into one of the starkest lines you'll find anywhere in population genetics. In West and Central Africa, this variant is close to fixation. Nearly everyone carries it, and you move to European or East Asian populations, and it nearly vanishes, disappears. And that gap isn't a difference in what the mutation does biologically. The broken GATA1 site behaves exactly the same way in anyone who carries it. What differs is simply how common that broken copy became in different populations. And that difference traces straight back to how much pressure vivax malaria put on each of those populations over time. So one base pair, one transcription factor losing its grip in exactly one cell type, and a trait now carried by hundreds of millions of people, tracing back to a battle with a parasite that's been fighting humans for a very long time. See you on the next one.